We are going to take polymers, complexes like proteins, and break them down into amino acids. That would be a catabolic reaction that releases energy. Does this sound familiar? Please tell me that sounds familiar. All right. Hey, guess what? The starches, the carbs, we're going to break them down into simple sugars. Does that sound familiar?
Yes.
Okay. It is important for you to know, just in general in this world, yes, you can break down fats for energy. Yes, you can break down proteins for energy. Oh, he's starving, he's breaking down his muscle for energy. Yes, that stuff really happens, but we're only going to focus on the breakdown of the carb. If you want to do the whole everything for all of metabolism, you would go next door into the micro lab and take it from micro. We're just doing the breakdown of the carbs, breakdown of glucose. Things I actually care the most about, I made big, not just red, I made them big. Yes, there's stuff about human weight and what the body uses for energy and all, but the big thing here that I care about is respiration and how you get energy out of food. We're going to talk about both aerobic, that means with oxygen, and anaerobic, which is what causes fermentation. Okay? Food is the source of energy, and it mentions the fact that when you see calories written on food, that's really a kilocalorie. That's 1,000 calories the way a chemist would talk about calories, the way a physicist would talk about calories. And I like the fact that they talk about here 1 gram of water raised by 1 degree Celsius. It requires an amount of energy that would be 1 calorie. They actually could have gone further than that, because the 1 gram of water will occupy a volume of 1 milliliter, and which the 1 milliliter is the same thing as 1 cubic centimeter. The whole flippin' metric system is based on water. Metabolism, you already know that you have anabolic and catabolic reactions, right? You already know that in anabolic you are adding molecules together. Now the one we really care about on the next test is the one we just did a whole PowerPoint on, adding together carbon dioxides to make sugars. Now, are you actually making glucose?
Yes.
Eventually, okay, but the first step, you're adding carbon dioxide to make G3Ps, right?
Yes.
Yeah, you add three times through the Calvin cycle to make a G3P, 3 times through the Calvin cycle to make a G3P, stick them together, you got your glucose. Good?
Yes.
All right, so that's an anabolic reaction. It needs energy. It gets the energy from the ATP, and it gets the ATP from the light reactions. All of this ultimately comes from the sun, anabolic from the sun. This chapter, we're going to deal with catabolic. We are cutting the big molecule apart and getting the energy out of its chemical bonds. It's an okay picture. I prefer this one because it emphasizes the idea that things like the carbon are used over and over and over again, go around and around in the atmosphere, in the ecosystem, but energy passes through. If you remember we had the picture of the cell and what happens if you cut off energy coming into the cell just dies, right? Whole ecosystems die if they don't have sunlight coming in. Almost every ecosystem on Earth is based on photosynthetic organisms at the bottom, autotrophs. Other ones, like bottom of a deep sea vent where there's no light, their autotrophs are going to be chemosynthetic. But almost everywhere on Earth, sunlight is where the energy comes from. So food's a source of energy. Food has macromolecules. We talked about that before. You understand that we can break down sugars for energy. We can break down amino acids for energy. We can break down fats for energy. We can break down nucleotides and recycle their parts, but there's not much energy in them. Which one of these are we actually going to emphasize? Are we actually going to go through all the steps of the breakdown of this chapter? No. Carbs. And in us, when we talked about carb loading, we talked about how you can store these carbohydrates, these simple sugars, these glucoses, you can hook them together in glycogen, right? When someone does carb loading before, a runner, a swimmer, they're going to go out and have pasta a day and a half, 2 days before their event. The reason why is because you put the actual sugars you're going to use in the muscles that are going to use them. This is how you hook them together temporarily to store them. If you eat if you eat more calories, more energy than you can use, you'll store them there temporarily. If you keep doing that and the muscles are full, then you store it as fat. That's the answer to the books harping on why are people fatter. Now we eat more calories than we used to. And what I already said, you can only burn the fat if you've used up all the glucose in the bloodstream. the glucose in the glycogen in the muscles. At that point, your body's like, hey, we have to burn something else. What do? You go for fat. All right, what I want you to know, the whole reason that we are eating food, we're trying to get energy in the form of ATP, adenosine triphosphate. That is what's used to power the cells. It has 3 negatively charged phosphates. Nick, do 3 negatively charged things want to hang out together?
No.
No, exactly. So you have 2 ADP, and then you try to bring this third phosphate. It's like, I don't want to be there. But there is a molecule that can, through force, stick them together. What molecule would that be?
ATP.
Excellent. So would you agree that ATP, when we break this bond and this phosphate comes off, it can release energy? So inside the cells, enzymes are going to break the bonds. That's all the more the book says. It's really light and fluffy about it. We're about to give you some serious detail. Same level of detail that we had with the chloroplasts. So the energy is going to be captured and released. It will be converted to make up ATP. So we're going to break apart a sugar, and every time we break it, we're going to capture the released energy. There'll be high energy electrons. We're going to absorb them. We're going to carry them over and use them to make ATP. Well, we'll use them in an electron transport chain to pump what? What do you think we pump? It has to power the ATP synthase. So are we going to pump? Hydrogen. Oh, this is important. I want you to know, you'll see this in today's lab. I want you to know that both producers and consumers do respiration. What that means is that plants have mitochondria too. Everybody, when they talk about photosynthesis, they talk about what, oh, look, the plant, and it does the photosynthesis. They forget that the plant also has mitochondria. The plant also does respiration. You need to know that. You need to know that they do both. We're now going to talk about the respiration, talk about the mitochondria, and there are 3 processes in the respiration, in the breakdown of sugar to get all the energy out of it that we can. The first is called glycolysis. This does not happen in the mitochondria. Write this down. This happens in the cytosol. Remember, cytoplasm is all the stuff between the nucleus and the plasma membrane. It's the liquid, the cytosol, and all the organelles in it, right? Cytosol is where glycolysis happens. It happens in the liquid of the cell. The Krebs and the oxidative that we're about to talk about happen in the mitochondria. Glycolysis does not need oxygen. Krebs needs oxygen to happen. Oxidative phosphorylation needs oxygen. That's what the oxidative means. Now, in the olden days, they used to call these glycolysis, Krebs, and the electron transport chain. The problem with that is it makes people think there are no other electron transport chains. We're trying to get away from that. The new term that people use is oxidative phosphorylation. And you're like, oh my God, that's so big and so many syllables. But it's simple. Oxidative means it uses oxygen. Phosphorylation means we're sticking a phosphate on. All it's talking about is taking 2 phosphorus ADP and sticking the third one on to make it ATP. You're phosphorylating the ADP to make it ATP. Good? Good? So, yeah, it's a long word, but I want you to know glycolysis outside of the mitochondria, no oxygen. Krebs inside uses oxygen. Oxidative phosphorylation inside the mitochondria uses oxygen. Good? Good? Hey Nick, which one of those 3 doesn't use oxygen?
The first one.
Excellent, excellent. And its name would be?
Glycolysis.
Yeah, sweet. I'm impressed. All right. So, aerobic respiration is a series of reactions. Aerobic means with oxygen. Okay? Series of reactions converting food energy into ATP. Aerobic with oxygen. Now, you don't need it for the glycolysis. There is something that I left out. I said you don't need oxygen for glycolysis. It's totally true. Glycolysis is, the name means lyse, break. So you're breaking sugar. You're taking a 6-carbon glucose and breaking into 2 3-carbon pyruvates. Good? But you can't move the pyruvates into Krebs unless you convert them to acetyl-CoA. And don't write it down now. You'll memorize that in a bit. It's on another slide. That pyruvate that has to be converted to acetyl-CoA, that requires oxygen. So if you want to be really technical, glycolysis, no oxygen. Krebs. Well, before we even talk about Krebs, you need to use an oxygen to get the stuff into Krebs. Then within Krebs, you need oxygens. And then you need an oxygen at the end of the oxidative phosphorylation. All right, good? So now let's look at it. Glucose is the most common source of energy for all organisms. It is the main energy. Your brain runs almost primarily on glucose, almost exclusively on glucose. Things like your heart, they can run on fatty acids just fine. They do that at rest. When you're not exercising hard, your heart runs off fatty acids. Brain runs on glucose. To the point that if you take all the glucose out of the blood, put it in the muscles, the brain stops working. That's what an insulin-induced coma is. Does that make sense? The brain doesn't have a backup. You would think that the most important organ in the body, you'd have a backup fuel. But we don't. That's a great design. All right, so aerobic respiration of glucose. Hey, well, I'm not going to repeat it. You can see you need to know the inputs and the outputs. That's true here. That's true in photosynthesis. And they are the opposite processes. Remember, in photosynthesis, we took in water, we took in carbon dioxide, we took in energy, and we made sugar and oxygen. Now we're going to take in sugars and oxygen, and we're going to get out energy, carbon dioxide, and water. Good? Good? The difference is that when we did photosynthesis and went that way, the energy was light. We're going this way, and the energy is going to be ATP, adenosine triphosphate. Good? Actually, there are a few other things made, like NADPH, NADH. We'll talk about those. There are 3 steps. Aerobic respiration requires oxygen. You got to know that. You got to know the word aerobic. And there are 3 steps. There is glycolysis that happens in the cytosol. There's the Krebs cycle. Yes, you can also call it the citric acid cycle because one of the chemicals in it is citric. I want you to know Krebs because it's easier. So citric acid cycle, Krebs cycle, and mitochondria require, this happens in the mitochondria and it needs oxygen. It's what they have drawn here. And this is a big deal because you're seeing the pyruvate go in. Actually, it turns into acetyl-CoA before it goes in. You'll need to know that. And as you go around, you're breaking it apart and giving off carbon dioxides. It's because you're breaking the chain, taking the carbons off. And how do you get rid of the carbons? Hook them to oxygen, breathe them out. Good? Good? And what we're doing when we break those bonds and get rid of the carbons, we're harvesting the high-energy electrons. We're going to keep and use the electrons. Those electrons are absorbed here. I'll explain how in just a second. This NADH is full of these high-energy electrons. It carries them over to, your book calls it the electron transport chain, but it's an electron transport chain. It is the long one. It is the one in oxidative phosphorylation. Good?
Yes.
All right, so 3 steps: glycolysis, Krebs, and I want you to call it oxidative phosphorylation, but the one they're calling the electron transport chain. Are you with me? All right. So know that the second 2 steps require oxygen. Know that they're in the mitochondria. All right. So the first part, I already said, it's named glycolysis, which means breaking of the glucose. It happens in the cytosol. You know this. You're taking 6 carbon glucose, breaking into 2, 3 carbon pyruvates. Good? Yes. Excellent. Moving on. All right. There's no oxygen required for this to happen. Oxygen is needed if you carry on in the breakdown of glucose. Good? So, you're going to treat this like a black box. You do not need to know the names of the steps. You do not need to know when you're breaking things. You do not need to know the names of the enzymes. If you were a bio major, I would throw all that rubbish at you. Okay? All I want you to know on this is that you do the 6 carbon glucose, break it into 2, 3 carbon pyruvates, and I want you to know, just like they say, it takes a little money to make money, it takes a little energy in order to make energy. We're going to burn up 2 ATP starting the process. We're going to get 4 ATP out. So there's a net gain of 2 ATP. And we also make some of these awesome NADHs that carry our high energy electrons. Good? You're only making a little in glycolysis. You're making 2. You make a bunch of these, a buttload of these in the Krebs cycle. That's what the Krebs cycle is for. The pyruvate will then enter the mitochondria. We have to convert it first. that it will go into steps 2 and 3, the Krebs and the electron transport chains. We also, as we do this, would you agree that we broke a bond between carbons here? Right? If you break a bond between carbons, you release high energy electrons. Those high energy electrons are socked up. They're used by the NAD+. We'll talk about that in a slide or two, but the whole point is that NAD+ becomes NAD once you pick up an electron to balance that plus. And then it picks up another electron, so it becomes NAD-. Well, how do you balance that out? You pick up a hydrogen. That's why it's NADH now. Does that make sense? It carries those off to oxidative phosphorylation, and then it lets go of the high energy electrons and the hydrogen, which is great because what are we going to use this high energy electrons for? To pump. Yes, very good, Alethea. You're going to use the high energy electrons to pump the hydrogen. Alethea's got it down. All right. So glycolysis doesn't use oxygen, and it's a nice thing, but it's not enough to keep us alive. There are a few things that don't need a lot of energy in this world. Like there's some bacteria that live on nothing but glycolysis. But most things in this world need more energy than that to get by. Does this make sense? You with me? All right. So since we can't get enough energy out of glycolysis, we've got to do something else. What we're going to do, we're going to go through 2 more steps. We're going to do Krebs, and that's going to require oxygen. We're going to do oxidative phosphorylation. It used to be called the electron transport chain, and that's going to require oxygen. Most of the ATP making happens at this last step. That is a test question. You've got to know it. All right.
You said it happens at the last step?
Yeah, the last step is where we make most of the ATP. All right, so the Krebs cycle, part 2 of breaking down sugar. Part 2 of cellular respiration. This is aerobic because it's using oxygen, because it's happening in the mitochondria. And this is why things like muscle cells that need lots of energy, they've got lots of mitochondria. Does that make sense? Think of your heart. You pump, always pumping, full of mitochondria. Also lots of blood flow to the spot because you got to deliver lots of oxygen to those mitochondria. Does this make sense? Not getting enough nod. Does this make sense?
No.
No?
As a nod.
Oh.
Giving you the nod you wanted.
Okay, thank you. All right, so the Krebs cycle part 2. We've got to turn the pyruvate. Where did the pyruvate come from? It's the last stage of? Yes, glycolysis. Very good. The pyruvate comes from the glycolysis. It's the output product of the glycolysis, and we're going to put it into the Krebs, but first we have to change it. We're going to turn this pyruvate into acetyl-CoA, and that needs oxygen. Pyruvate has 3 carbons, and we're going to break carbons off. And what do we do with them when we break carbons off? We breathe them out. That's why we need oxygen. All right? So, and before we get to the slide, the picture of Krebs, I want you to know that Krebs does make a little ATP, and our ultimate goal is to make ATP. But what it makes is a lot of this NADH that's carrying the high energy electrons, that's carrying hydrogens. Good? All right. And it'll make a little FADH2 that's also a carrier molecule. So here we have it. Krebs cycle, turning the pyruvate into the acetyl-CoA requires an oxygen. Why? Because we take these 3 carbons, we break one off, we hook it to oxygens, and we breathe it out. This is the reason why we say that you make carbon dioxide, you breathe out carbon dioxide. The reason you need to take in oxygen and breathe out carbon dioxide is because every time we break a carbon off, we breathe it out by hooking it to an oxygen. Yes?
This might be a little off topic. What is the FADH2? Like, what is that?
It is another carrier, it works the same way, as another carrier of high energy electrons and hydrogens off to the electron transport chain. All of this, the ATP, that's just raw, that's just the energy. That's what we want to make, and yay, we made some. But the whole point of NADH, FADH2 is to carry electrons off to put them through an electron transport chain. Good? Good? All right. So we made our acetyl-CoA. We had a 3-carbon pyruvate, we broke the carbon off, we breathed it out, we now have a 2-carbon acetyl-CoA. It goes into the citric acid cycle. So we have this 4-carbon one, it grabs the 2-carbon one. What does that make? 6. It's not complicated. We have a 6-carbon thing. And no, you don't have to memorize the numbers and all the steps. Relax. I'm just wanting you to get the concept. So we have this 4-carbon oxaloacetic acid, and it picks up an acetyl-CoA, and bam, we got citric acid. Well, what do we do with it? Right away, we take this side chain, this 1-carbon side chain, break it off, breathe it out. That's why we need an oxygen there. That's why we create a carbon dioxide there. Well, when we do that, we're going to release high energy electrons. The NAD plus absorbs those high energy electrons, 2 of them, which then has to pick up a hydrogen to balance. That's why it's NADH. And it carries the hydrogen off. It carries the electrons off to use them in making of ATP. We can talk about ATP synthase some more if that helps.
The chair is really helping it stick in the brain.
And what does the chair represent?
It's ATP synthase.
Exactly.
See,
It's and how does it represent it? A molecule spins, right.
If I remember nothing else in this class.
I remember it spins and it's up.
All right. So, and what is it that causes the ATP synthase to spin? The movement of? Yeah, say it louder. I heard it. Hydrogen ions. Hydrogen ions. So what we're doing here, NAD plus picks up one of these high energy electrons that's released from the broken carbon bond, and it becomes NAD. You know, the electron balances it out. Then it picks up another one, and that makes it NAD minus. Well, we don't want to be that way, so what we do, we pick up a hydrogen to balance that out. And that's great because then we carry that, we're now NADH, we carry all of that stuff over to the electron transport chain of oxidative phosphorylation. Well, what are we going to do? We're going to use these to power the electron transport chain, right? But what are we going to do with this? Well, we brought one of the things along that we have to pump. It's all good, right? We deliver everything right there to where the action happens. Make sense? Make sense? All right. So you need to know that NAD plus picks up electrons and hydrogen. They're released from catabolic reactions, as long as we're cutting the bonds between carbons. We do that, we release high energy electrons, and we're going to absorb them and use them. Good? All right, so the Krebs cycle, cellular metabolism, releases carbon dioxide. This is why when we breathe out carbon dioxide. Good? Oxygen is needed at the start because we're going to turn the pyruvate into acetyl-CoA. And every time we break carbons, the bond between carbons, we're going to capture, using NAD+, capture the hydrogens that are released, capture the high energy electrons that are released, and we're going to use them later on in the 3rd step of glycolysis to make ATP. To pump hydrogen ions that will then be put through the spinny chair, right? They'll be put through the whole idea is that we are pumping hydrogen ions that will then be used in the Carter-friendly method of ATP synthase. You guys understand that the hydrogen powers the spinning of the ATP synthase, right? You have to know that hydrogen powers ATP synthase. So, energy molecules from Krebs. In Krebs, we're going to make a little bit of ATP. We're going to make a little bit of FADH2, but the big thing Krebs does is make lots of NADH. Good? And that NADH has the hydrogens that we need to pump, and it has the electrons to pump them. Are we good?
Yes.
All right. And so we're going to use these to make ATP. We don't waste energy, we break carbon bond. All right. So Melvin, we're here, we're at part 3. That looks like a big word, but do you understand it? It's oxidative because it requires oxygen. It phosphorylates things. It's phosphorylation because we're shoving another phosphate onto the ADP. Are we good? Yeah. All right. This is where this happens in the mitochondria. It needs oxygen. You probably guessed from the name. All right. But it's where most ATP comes from. That's in bold, in italics, in all caps, and in red.
Was it? Yeah.
Oh, with 4 exclamation marks after.
Oh yes, with 4 exclamation marks. It's real important that you know the 3rd step is where you make most ATP. It's also real important, I don't have a slide for it, that you know that in both the chloroplasts and the mitochondria you have similar things happening. What powers the ATP synthase in chloroplasts? Yeah, say it loud. The hydrogen ions, the movement of hydrogen ions. What powers the ATP synthase inside of mitochondria? Hydrogen ions. The movement of hydrogen ions powers ATP synthase. That's true whether we're doing the light reactions of photosynthesis or we're doing the last part of the mitochondria, the last part of cellular respiration. Good? ATP synthase is powered by hydrogen. Hydrogen ions. Or if you want to be nerdy, you can call them protons, remember. All right, so oxidative phosphorylation, you need to know that it is the last part, the third part. You need to know that it is the part that makes the ATP.
Yes.
Good, it makes most of the ATP. It does this by taking electrons that were released in glycolysis, electrons and hydrogen from glycolysis, electrons and hydrogens from Krebs, and carrying them to the inner membrane of the mitochondria. This inner membrane, remember, mitochondria has a double membrane system. We have an outer membrane and we have an inner membrane. The inner membrane has the electron transport chain. The inner membrane has the ATP synthase. We're going to be pumping hydrogen from the middle of the mitochondria into this space between those membranes. And then it will flow back to the middle through the only method it has to go, a hole through the ATP synthase. Right? Right? And what does that do to the ATP synthase, Carter?
What does it do to the ATP synthase?
When hydrogen flows through it, what does it do?
I'm not sure.
It makes it spin.
Synthase.
You really didn't remember that?
That's the chair thing.
I'm traumatized.
I want you to remember
Block it
that
out,
hydrogen
detergents.
makes ATP synthase spin.
Okay.
Good. All right, so then we have oxygen. You have to know about oxygen because it's oxidative phosphorylation. What are we using oxygen for? It is an electron receptor. We'll explain that in a second. This process makes most of the ATP. I said that twice. So here are the pictures. We are going to be pumping hydrogen across, this is the middle of the mitochondria. This is the intermembrane space, the space between the outer and the inner layer of the mitochondria, outer and inner membrane. You need to know that's where we store hydrogen. You need to know that hydrogen is stored between the outer and the inner membrane. We call that the intermembrane space. You need to know that we are pumping the hydrogen from the middle of the mitochondria to the intermembrane space. And we're pumping that because we're using energy from high energy electrons. This is the long electron transport chain. So we have a high energy electron, we get it from the NADH. I want you to know that. We get it from the NADH, and that energy electron, it goes from this protein to this protein to this protein to this protein. As it does, you see that little diagonal? It's because it's going lower energy, lower energy, lower energy. Good?
Okay, I got that hydrogen
Yep.
is stored between the outer and inner membrane space. What was the second one you just said?
That it's being pumped there by the electron transport chain, by an electron transport chain, the one in oxidative phosphorylation. Good?
So basically it's going in and out multiple times to the
Right,
next level.
right. Which is the same thing as in the chloroplast. It comes from the stroma into the thylakoid, it flows back into the stroma. It comes stroma to the thylakoid, it flows back into the stroma.
Each time it's losing energy.
Well, no. When you said it, I thought you were talking about the hydrogen ions. The electron is going from high energy to low.
Oh, okay.
That's this blue E negative.
Right.
Going high energy, less energy, less energy, less energy. When it's all used up and has no energy, who wants an unwanted, unloved, doesn't even have any energy left electron? Oxygen wants the unused electron, the unwanted, doesn't have any energy left, because oxygen is electronegative, remember? That means oxygen wants electrons, oxygen wants to become negatively charged. Sorry, if the electron has energy and nobody else wants it, you still want that low energy electron because you have an undying love for the electron.
I do.
All right, and that's why we use it as the receptor at the end of the chain. Good? All right, we call it oxidative phosphorylation. It is the place where we make most of the ATP. It is the last stage. It happens in these folds. We call those folds cristae, but it's the inner membrane of the mitochondria. You would agree, once we have lots of hydrogens here, they can only flow through one way. It is the spinny chair. It is this molecule. ATP synthase. So if you're ever wondering yourself, what's the name of that enzyme that makes ATP? Just say it in science speak. What's the name of that enzyme that makes ATP? It's ATP synthase. All right. So it's the third stage. You know where it happens. It happens in the inner membrane. I want you to notice the inner and not the outer. Hint, hint, hint. Okay, so This uses oxygen at the end of the chain, and doing this, we produce ATP in large amounts. We made a little bit in glycolysis, we made a little bit in Krebs, but remember glycolysis? We used 2, we made 4, so we got a gain of 2, right? We made 2 of them in Krebs, yeah, whatever. We break down a glucose and we pass it along. The electron transport chain is where all the action actually happens, the last part. So we take any high energy molecules that absorb electrons, absorb hydrogens, we carry them off to oxidative phosphorylation, and we use them to make ATP. Are we good?
36, right?
36 total if you are 100% efficient and you don't take any of the molecules off to use for other things. That's why I said 30, because a lot of times you siphon off some of those things to make fatty acids and do other fun things. Protons move from high to low concentration. Protons is another name for hydrogen ions. So we've got a lot of them here in the intermembrane space, and they flow through the ATP synthase. And when that happens, it powers the turning of an ADP with a phosphate. You phosphorylate it, you slam that phosphate on there, you make an ATP. Moving protons, moving hydrogen ions are the thing that power ATP synthase, right? All right, so electrons from this electron transfer chain go to oxygen, right, Zach? Oxygen picks up those unwanted electrons, and when this happens, what are you going to do with them? Well, you have air you breathe in, right? That's where you get your oxygen. But they pick up an electron. Well, now they don't want to make this diatomic bond anymore. You have an O negative by itself, and wow, that's like dangerous. That's a free radical that could do horrible things to you. So what do? Well, you have a lot of hydrogens here that have been passing through, right? So you hook the hydrogens to that oxygen and you make H2O. And you do it twice because you have an O2 you broke apart, right? O2 comes along and one of those oxygens picks up an electron and then it says, oh, I don't need that other oxygen. And now you have 2 dangerous, unstable oxygens. What do? Well, they pick up the hydrogens that are on the inside. That's good because that helps keep the hydrogen level here low, helps keep the hydrogen level here high, right? They flow through and they're immediately joined up into water. Remember we said one of the waste products, we breathe out a little bit of water? That's why. Does this make sense? Yep. Hey, cellular respiration, the total yield. Okay, you're right, 36 if you did things perfectly. Most of the time we get 30 to 32.
So 30-ish.
What? Yeah, exactly. That's why I said 30. So cellular respiration, 3 steps. Glycolysis, we don't need oxygen. Krebs, we need oxygen. Oxidative, we need oxygen. If we go from glycolysis to Krebs, we need oxygen because we have to make the Megan what? If we go from glycolysis, if we take a pyruvate from the end of glycolysis, and we're going to put it into Krebs, we have to turn it into what? Looking for an A word. Yes! Very good, Sean. Acetyl-CoA. All right. So, total yield 30 to 32. Now, it's true that we can burn other things. You can burn fats for energy, you can burn amino acids for energy. Fats will have more energy in them than amino acids will because they got more electrons. Remember, the electron transport chain of oxidative is where we make most of our ATP. Yeah, it's all good. So anyway, sometimes oxygen consumption you can use more oxygen than you need, right?
Yeah.
If you use, you can need more oxygen than you have. That's what I should have said. If you don't have enough oxygen, there are still ways to go on because glycolysis doesn't need oxygen, right? Yeah. So you could still, if you don't have enough oxygen, you can still do glycolysis. Tell you why you make lactic acid or alcohol. We would make lactic acid when we're running without oxygen. If you take yeast and you run them without enough oxygen, they're going to make alcohol. Anybody who likes to drink beer, thank it. Thank the yeast. You remember this. You remember our black box? I said we weren't going to make you know the names of anything or the individual steps. I'm not lying, we're still not. But I do want you to understand that it makes NADH, right? And we already talked about what that's used for. It's used to carry electrons and hydrogen off to use an oxidative. Good, good. Well, here, if you run out of NAD+, you can't make NADH. If you can't make NADH, you can't go past that step. Exactly. You can't finish the glycolysis. And that means you never get down here to make the ATPs. Then you have no method of getting energy and you're screwed. So what do? Your body says, well, if we can turn the NADHs back into NAD+, then we can continue down this chain and get the ATPs out that we need. So what do? It's pretty simple. You take the pyruvate at the end, and you attack it with the NADH, and the pyruvate will rip the electrons and the hydrogen off, turning the NADH back into NAD+, and then you can keep going and make ATP. Because when you're without oxygen, the only ATP you get is the ATP you get from glycolysis. Sorry, that is an exaggeration. Like I said, you can break down fats, you can break down proteins, but in terms of breaking down sugars, if you have no oxygen, all you've got is glycolysis, so you want to make this happen. We're going to turn the pyruvate into something else, and the reason we're going to do it is we want to turn this NADH back into NAD+, so we can keep going, keep doing glycolysis. In us, that pyruvate is turned into lactic acid. In yeast, it's turned into alcohol. You know about lactic acid. You've felt lactic acid before. If you've ever been exercising and the coach, you're running and you're breathing hard and you can't take it anymore, you can't breathe any harder, your muscles start to ache and the coach yells annoying things like, feel the burn! That's what they're talking about. That pain in your legs, that's lactic acid building up. Okay? Now, it's kind of really clever the way this works. If you build up lots of that lactic acid, that area of the body is getting acidic. Would you agree? Well, guess what? Hemoglobin, which carries the oxygen from the lungs to the tissues, it dumps, well, I'll use the science words in a second, but it dumps oxygen better in places where it's acidic. So the places that need the oxygen the most are most likely to get it. A nerd way of saying that would be that acidic area, hemoglobin has a lower affinity for oxygen in acidic conditions. Good? Good? Makes sense? All right. So when we have fermentation, yeast makes ethanol, yay! In us, our overtaxed muscles, they make lactic acid. Here's the picture of it happening. We take our glycolysis, and we're going through glycolysis, and the only way we can keep going is if we have more NAD+. Like we made our ATPs, well, how do we get our NAD+ back? We take the pyruvate and we attack it with the NADH. That turns the NADH back into NAD+, and then you can keep going. In yeast, that makes ethanol, and us, it makes lactic acid. Good? Yeah. So, pretty simple. Pretty simple.
That's okay. I've been bitten. Ow, he has teeth. Ow, ow, ow.